Hi, my name is Thomas, and in this presentation, I'm going to tell you about how to discover and validate TCR candidates using MHC and TCR Dextramer reagents as provided by Imidex. I will be talking about some of the challenges in detecting antigen-specific T cells and some of the possible solutions to that, what you can do in the terms of TCR discovery, how to characterize and validate TCRs once they've been found, and how to make these TCRs and turn them into relevant reagents and solutions. I will round off the presentation with a couple of examples demonstrating the use of TCR-based reagents. T cells play a central role in the immune system as master regulators of immunity, as well as effector cells carrying out essential functions to directly combat disease. And this also means that understanding T cells and the targets they respond to is absolutely key to understanding immune function. And it all begins with our ability to detect T cells in an antigen-specific manner. So what is the challenge? The challenge is essentially one of affinity. And detecting T cells is not trivial. When T cells develop in the thymus, they undergo a two-step selection process. The first so-called positive selection step ensures that fully developed T cells carry a functional T cell receptor, while the subsequent negative selection ensures that the T cells are not reactive to self-epitopes. The resulting T cell repertoire that is released continuously into circulation is therefore biased towards recognizing mostly foreign peptides. The process of positive and negative selection relies on the affinity of the TCR towards its target. And this also means that the more foreign-looking an epitope, the more likely it is to find a reactive T cell, and the higher the affinity of the T cell for the target will generally be. On the other hand, the more self-similar an epitope is, the less likely it is to find a reactive T cell, and most such T cells will exhibit very low affinity towards their target. If we translate this phenomenon to the different types of diseases, we see that antipathogen TCRs tend to be of higher affinity, while autoreactive TCRs tend to be of very low affinity. And anti-cancer T cells often target self-epitopes or some very similar, self-similar neoepitopes, and thus they also typically exhibit modest to low affinity for their targets. If we take a closer look at the interaction between a TCR and an MHC peptide complex, we find that the interaction is generally weak compared to, for instance, antibody-antigen interactions. While antibodies typically exhibit dissociation constants in the nanomolar or even subnanomolar range, TCR-MHC peptide interactions are typically in the micromolar to millimolar range. And this means that even good TCR-MHC peptide interactions are short-lived on a monomer level, with half-lives down to the order of seconds. In vivo, this apparent limitation is overcome by having several TCRs on the T cell interact with several MHC complexes on the target cell at the same time. And this increases both the strength and the duration of the interaction between the T cell and the target. And it allows for the activation of the T cell and all the subsequent effector functions to kick in. So by multimerizing this interaction, we can increase the effective affinity, also known as the avidity. And this multimerization allows us to increase the interaction time from mere seconds up to minutes or even hours. Traditionally, people have been making MHC multimers using streptavidin to create tetramers. Tetramers of MHC complexes will typically bind to T cells with half-lives on the order of minutes. But it's possible to go to much higher multimers, such as dextramers, that will give you half-lives of the reagent on the T cell surface on the order of minutes up to hours. By going to these high-valency reagents, it's also possible to detect weaker interactions than is possible with the low-valency reagents. And you can actually find some of the weakest interactions, such as those relevant in the context of cancer and autoimmunity. The use of immunotherapy to combat disease, in particular cancer, has shown a lot of promise, as evidenced by lots of recent results in the field. And one of the most recent flavors of immunotherapy with broad applicability uses what's known as TCR T-cell therapy. And here cancer-specific T-cells are identified, and following one of several possible optimization or engineering routes, the TCRs they carry can be transformed into T-cell or TCR-based therapy. Building a TCR T-cell therapy follows the steps outlined here. First, you have discovery, where you identify the relevant epitopes and TCRs recognizing these epitopes. Then follows a phase where you characterize the identified T-cell receptors to ensure that they have the right properties in the context of your disease. Then these TCRs can be introduced into, for instance, a T-cell to create a potential therapy, and then this needs to undergo extensive clinical trials and testing before being released as a treatment in humans. So let's have a closer look at some of these steps in the process. First, target discovery. If we take a look at the immune repertoire, it is very, very large. So finding the right TCR for a given application is essentially a question of finding the needle in the haystack. If we look at the human T-cell repertoire, it's been estimated that at any given time, the number of different TCRs present on T-cells circulating in the body is on the order of tens of millions. And each of those TCRs in its own right can detect maybe up to millions of variations of a given peptide sequence. If we look at the variability on the disease side, so in the context of cancer, for instance, not only is there variation from patient to patient, there's also variation within the same cancer, within the same patient, and the variations might even change over time. So all of this combined variability means that you need some sort of high throughput technology to find the right TCR for the right application. One way of doing this initial step of finding the right epitope is illustrated here. Here we've taken MHC multimers with different specificities. We've attached DNA barcodes to each of them, and we've then created a library of different reagents that we can pool. This library of reagents, in the context of Imidex, it's called TCR Dextramer reagents. This library can be used to stain a pool of cells, be them PBMCs or tumor samples. Once stained, the cells that bind these reagents can be sorted based on the fluorescent markers present on these reagents. And then the DNA barcodes from these sorted cells can be amplified in PCR and detected using deep sequencing. And this essentially allows you to identify which of the initial MHC peptide specificities are recognized by the pool of T cells present in this sample. So this essentially allows you to do epitope discovery, and you can go up to hundreds. And in the context of this paper mentioned here, they've tried to use more than 1,000 different specificities at one time to identify relevant epitopes. If you want to understand the TCRs more deeply, one way of doing it is illustrated here. We can essentially do usually workflow similar to the one in the previous slide. We can create MHC peptide reagents. We label them individually with DNA barcodes, and we use a pool of these reagents to stain T cells. We can sort these T cells, and after sorting, we can partition them into single cell reactions using either the BD Rhapsody or the 10x Chromium system. This allows us to do single cell reactions that allows us to identify which markers are present on the surface of each cell, what the sequence is of the T cell receptors carried by each individual T cell, and we can have a look at the gene expression. And all of this can be combined to give you an image of the specificity and the phenotype of each and every T cell. Here's an example where we've used the single cell sequencing setup. Here we've taken a pool of PBMCs, we've stimulated them with a couple of viral model epitopes, one for tetanus toxoid, one for an EBV epitope. Following peptide simulation, the PBMCs have been stained with MHC peptide dextramer reagents specific for these two peptides. And we can see here that we can specifically identify those T cells. The T cells have then been subsequently been sequenced, and we also now have the sequences of their T cell receptors, which could be taken further to use in therapies or other relevant applications. So, using these DNA barcodes on these on MHC multimers, such as Dextramer reagents, it's possible to do massive multiplexing of your epitopes, either to do epitope discovery or to go one step further and have a look at the phenotypes of the individual T cells and the T cell receptor sequences that they carry. So, at this point, you will have identified relevant T cells of the relevant phenotype and the T cell receptor sequences that they carry. So the next step is validating the properties of the TCRs recognized. In the previous slides, I showed you how to detect antigen-specific T cells using the MHC Dextramer reagents. And it's possible to sort of flip this question around and then take those TCR sequences identified, create the TCRs, create a multimer with the TCRs, and then use those to detect the antigen-presenting cells. Essentially, a symmetrical setup to the detection of T cells. If you have a TCR sequence, you can create the protein, and once you have the protein as a monomer, the TCR monomer, you can use this monomer to test for cross-reactivity and binding strength. Both of these are very important to understand if the TCR is to be used for any kind of therapeutic purpose. The cross-reactivity, of course, to avoid any kind of unwanted reactivity of a final treatment, and the binding strength to ensure that the TCR is of sufficient strength to give you the efficacy of the treatment in question. Now, making a TCR is simple enough, but one of the important things in the production of a TCR is to ensure that it also behaves the way you think it does. So we've come up with a way of producing TCRs so that we can validate the TCRs and ensure that they actually bind the target that we think they do. Once we created and purified these TCRs, we can assemble them into TCR Dextramer reagents, and we can then use these reagents for their final purpose following a number of quality control steps. Here's a quick example of how the production looks. The individual TCR chains are produced in bacteria, and the TCR is subsequently refolded and biotinylated. After purification, the TCR, as you see here, this small peak here, the TCR is tested for purity. And then once we have ensured that the TCR is pure, we need to go one step further and test for the functionality of the TCR. To do this, we've developed an assay that sort of mimics the situation of the TCR Dextramer binding to MHC peptide complexes on cells. But instead of using cells, we've introduced beads that we load up with MHC monomers, both cognate for the TCR and unrelated specificities. And in this kind of assay, we can ensure that the TCR Dextramer reagents bind specifically only to the cognate peptide MHC and not unrelated MHCs. And in this way, we ensure that the TCR is functional. Now, I just want to show you a couple of examples where we've used TCR Dextramer reagents to stain cells. In this first case, we've looked at the impact of the affinity of the TCR versus that of the peptide MHC. And we've taken 3 different TCRs, all targeting the same epitope, the NY-ESO1 peptide. This peptide is bound to the HLA-A02, and the 3 different TCRs exhibit very different affinities. TCR-A has an affinity of about 15 micromolar, dissociation constant, And then there's one affinity optimized version with an affinity about 1 nanomolar, and then an even more optimized version with an affinity down to 48 picomolar. If we create such TCRs and stick them on dextran backbones and stain peptide pulsed cells, we can see that they all are able to detect the MHC complex on the cells, but they do so with different signal intensity. The high affinity TCR, TCRC in this plot, gives much higher signal on this noise, but even the wild type TCR of modest affinity is actually able to detect the peptide on the surface of these peptide pulse cells. Here's another example where we've taken a non-classical MHC as a target and a TCR targeting this HLA-E molecule. Again, we've tested the TCR against the HLA-E complex against its native beats, and we see that it binds specifically to those and not to an unrelated A2 peptide MHC complex. And if we flip the question and we ask whether the HLA-E complex is specifically recognized, we also see that the A2 TCR specifically recognizes only its cognate complex and not the unrelated HLA-E complex. So we can say that we've been able to produce both of these TCRs, and we can show that they are specific for their cognate targets and don't bind to other unrelated MHC specificities. So just to give you an overview, we've now been through the initial epitope discovery. We've talked a bit about TCR validation and characterization, and the next step in the production of a TCR T-cell based therapy would be to introduce the TCR into cells and go for initial testing of the cell lines and then into clinical trials. So to summarize, the initial steps of TCR discovery typically start with epitope discovery, but following the epitope discovery, you can start looking into specific T cells and the receptor sequences that they carry. And at this stage, it becomes very important to make sure that the TCR is properly validated so that it exhibits the right affinity for your purpose, as well as the right specificity to avoid unwanted cross-reactivity. Once a TCR has been validated, the TCR can also be validated in a more biological setting, so we can examine the cross-reactivity of a TCR with this T-cell receptor, a T-cell with this TCR introduced. We can look at the cross-reactivity of the cellular therapy, and this can be done using MHC multimers, as you do for instance, epitope discovery. Reagents like this can also be used to isolate T cells infused with T cells with the right TCR introduced into them. And finally, the TCR Dextramer reagent can be used for the antigen sending cells, essentially the target cells, to assess whether the target cells carry the epitope of interest. Once the initial characterization of these TCR T cells has been carried out successfully, before being released for testing in humans, it's important to be able to characterize the T cells both in terms of identity and purity, but also in terms of specificity to ensure that you have a therapy of the right properties. And then finally, this can move into clinical trials if all the previous steps have been successfully completed. So thanks for listening.